So welcome everyone to the Population Dynamics virtual seminars. This time we have Michael Manhart. I will share my screen just to give you an idea of. I share everything, and I hope you can see the slide. So Michael Manhart from Rutgers University. I think he recently moved there. And just a few lines regarding the background of Michael. He got a degree in physics and mathematics at Stanford, a PhD in physics at Rutgers. Then he went to postdoc at Harvard, junior group leader in Switzerland, ATH, Zurich, and now is assistant professor at Rutgers University. So he has several interests actually, specifically in quantitative biology, ecology, evolution of microbial communities, with specific interest in evolution of microbial growth dynamics. And he also likes to inspect the effect of ecological interaction on adaptation in microbial communities. He would like to answer some questions with the research. What are the fundamental processes of evolution, and what are the physiological and ecological traits of microbial communities? So probably today he will give us some insight on some of that. With this, and I will stop sharing my screen and give the stage to Michael. So first of all, thank you for being here, and now up to you. You are muted.
Okay. Now can you hear me?
Yes.
Okay, good. Thank you so much, Marco, for the very kind introduction, and thanks for inviting me here to the Population Dynamics seminar, and thanks to everyone for coming. So today I want to talk about a few different areas of our work, actually, kind of already highlighted by Marco there in the introduction, that I would say are kind of broadly about the ecology and evolution of microbial growth dynamics. Let's see, okay. And you can still see my pointer?
Yeah, we can.
Okay, good. So broadly speaking, our lab is interested in quantitative evolutionary microbiology. And what that means to us is how evolutionary processes shape microbial communities. And so microbial communities are, as probably many of you here in the audience know, one of the most important topics in certainly microbiology, but I would say even in biology more broadly currently. And a major reason for their importance, of course, is their role in human health, not only disease, but also healthy functions of the human body, as well as really every natural environment that we know of, from the oceans to the soil, as well as many areas of biotechnology. But of course, these microbial communities are not static entities, but of course, they are driven by these fundamental processes of evolution, like mutation, selection, and genetic drift. And one of the main focuses of our lab is to understand how these evolutionary processes shape properties of these microbial communities, but also on shorter time scales, how properties of these microbial communities provide constraints for those underlying evolutionary processes. So for instance, they determine population sizes that affect things like mutations and select genetic drift. And in terms of the properties of microbial communities we're interested in, there are many things here that are, of course, important, but the ones that we're particularly focused on and that I'll be touching on today are especially growth dynamics of these communities, resource consumption, and if there's time at the end, a little bit about interactions between different kinds of cells. And the work I'm going to talk about today will start with some things from a little while ago when I was a postdoc. And so that was work done with my postdoc advisor, Eugene Shaknovich, and another postdoc in the group, Bharat Edkar, and also in collaboration with Ariel Admir and his current postdoc at the time, Jay Lin. That was funded by the NIH. And then more recently, the work that I'll be talking about was work that was started when I was in Switzerland. And a lot of that was done by an outstanding PhD student who defended just a couple months ago, Eustace Fink, and my collaborator there, Noel Held, who's moving to USC this year, as well as some excellent master's students and research assistants, Ashwin, Donat, and Anastasia. And that work was funded by the Swiss National Science Foundation. So I'd like to give you a broad outline of the questions I want to try to address today. So the first thing I want to talk about is how does non-steady state growth of microbial populations affect their evolution? And then after that, I want to talk about how the microbial growth response to a resource can evolve to be commensurate with that resource's environmental availability. And then finally, I think pending time, I want to talk about whether microbial growth can be co-limited by multiple resources simultaneously. And since I know these talks can kind of, go on for a while and you're all very busy, I want to just kind of give you the answers to these questions right away, at least as far as I'm concerned. So how does non-steady state growth affect evolution? Well, what I'll show you is that growth trade-offs can produce complex selection forces such as non-transitive and higher order effects. And then does microbial growth to a resource respond or evolve to be commensurate with that resource's environmental availability? And the answer to this, as it often is in biology when you ask a simple question, is not necessarily, and it turns out to depend on subtle details of population dynamics. And then finally, is microbial growth co-limited by multiple resources? We believe the answer is likely yes in many cases. And we think one major reason this could be the case is because evolution causes it to be so. So now let's get into a little more detail about these individual questions. So for me, one of the major interesting questions that I think we really need to grapple with more in this field now is what does the growth of microbial populations in natural environments actually look like? So obviously we've learned a lot about this in laboratory environments, but actually probing this in nature is, of course, the thing probably almost all of us are interested in. But of course, it's also a lot harder. And so I think from sort of the theory side, it's useful to sort of categorize some, fundamentally different possibilities for this before we really, think too much about data, for instance. And so one possibility for what this growth could look like is that if you just look at the log abundance over time, you could just have something that basically goes up at a constant rate. And of course, this is classic steady state growth. But the problem is that in nature, of course, this would be asymptotically infeasible. Things can't grow exponentially forever, or else we'd be drowning in microbes all the time, which we know we're not. And of course, there's also the opposite case here where you have steady state decay where things are just going down at some constant rate. And we know that's also not true asymptotically because otherwise there would be no microbes. So we can kind of rule that out. So a second possibility is that you could just have completely constant abundance over time. And I call this the chemostat-like regime because this is like what people would try to achieve in a chemostat in the laboratory. But the challenge with this, as anyone who's actually tried to run a chemostat probably knows, is that you have to balance birth and death at all times. And that's a very, fine-tuned arrangement of this system. And so that's also, I think, unlikely to be achieved realistically in natural environments either. So the third possibility is where instead of having sort of completely constant log abundance, you have constant log abundance over sort of long periods of times. But over short periods of times, there's some sort of cycling or fluctuations around that. And I call this regime the batch-like regime because this is something like when we grow batch cultures in the lab, this is something what they look like, where we give them maybe some supply of resources, they grow for a while until they run out of those resources, then maybe they sit there or they die a little bit. And then eventually they sort of are diluted down to some bottleneck size until they get new resources and can grow again. And the hypothesis that I think is very likely to be the case is that microbes are mostly growing in this sort of batch-like regime in nature. And I think this is important because this is, a non-steady state form of growth compared to like this first one here, which is just a constant, positive rate, or this one here, which is, a constant, zero net rate. And so the question then is, if that's true, growing in this out of steady state batch-like regime, how does that affect the ecology and evolution of microbes? So I won't be talking today about sort of evidence for this batch-like regime. That's a very difficult problem, and I'm honestly not sure there is much evidence for it right now. But I want to focus on just the consequences of that for ecology and evolution. So when I first started thinking about this, when I was a postdoc, one of the first things I thought would be useful to do was to start with a very simple model of these kind of growth dynamics and to see what could we learn just from some of the most basic ingredients of these growth dynamics. And so I think one of the simplest ways you can characterize this growth is by saying that you have some population, it's exposed to a supply of a resource, and at first there might be some lag time where growth doesn't occur, and then after that time has passed, growth begins at some constant exponential rate, and then eventually the population runs out of the resource and they reach some saturation phase. And so you have sort of these three-phase dynamics that would give you, this sort of classic sigmoidal growth that we often see in the laboratory. And so this would be characterized by three different traits. There's the lag time, a maximum growth rate, and then some resource efficiency or yield that determines when saturation occurs. And being interested in ecology and evolution, of course, we're interested in having multiple strains or species simultaneously competing under these conditions. And in particular, we're interested in what happens over longer timescales of these dynamics, not just sort of one of these cycles, but many cycles. And so you can imagine looking at how the absolute abundance of these strains or species would vary over many cycles, where they're sort of given their resources, they grow for a while, and they run out, and then at some time later, they're diluted down, and then they grow again, and then they keep doing this over many cycles. And as you can see, after just a few cycles, you start to really amplify even small differences between different strains or species. And especially from the evolutionary side, we really care about those differences. And so it's often easiest to look at not the absolute abundance, which of course goes through these more complicated cycles, but the relative abundance. And so like in this case, these blue and red strains start at 50-50, but then you can see after just a few cycles, they very quickly diverge. And after 4 cycles, the blue one has essentially, fixed in this population compared to the red one. And so this is the level of the dynamics that, especially from the evolutionary side, is often most important. So it turns out that just under this very sort of simple model of dynamics, you can actually get incredibly rich behavior of microbial populations. And sort of the argument here is that this is just because of sort of the non-steady state nature of this. And so to give you kind of the null expectation here, I think the best way to think about this is to imagine you have 3 strains, blue, orange, and green, and they grow according to these dynamics that I just showed you. And now what you can do is you can put together all pairs of these strains and look at how they behave in pairs, and then see how that corresponds to how they behave as a trio. And so, for example, in the simplest case, if you put all the pairs together, you'll probably find that one of these pairs just beats the other 2. So you can see how green will beat orange head-to-head, and then green will beat blue head-to-head. Again, these are those relative abundance over time plots. So these represent many growth cycles, not just one. But then when you put all 3 together, well, you see green beat orange and blue. So you expect that green will probably also beat orange and blue altogether. And in the simple case, that's what happens. But it turns out that under these dynamics that I showed you, you can violate this expectation in a number of different ways. And so, for example, one way is that even though head-to-head, green beats orange and green beats blue, you can find that when you put them all together, actually the green ends up losing, and the orange and the blue sort of collectively are able to suppress it. And in ecology, this would be considered an example of a higher order interaction, where the presence of a third strain, like say the orange one, has modified the interaction between the green and the blue compared to what they were going to do when they were just the 2 of them by themselves. Another thing that can happen is that when you look at all these pairwise competitions, there actually isn't a single champion of all of them. So orange beats blue and green beats orange, but then blue beats green. So you have sort of this like cycle or rock, paper, scissors like interaction between the strains. And then what that means is that it's very hard to predict what's going to happen at all in the 3 strain competition. And this was, as an example of in evolution, what would be called non-transitive selection. So there's not an underlying sort of scalar fitness landscape that determines these dynamics. And then finally, you can also have multi-stability here, where the outcome of this competition actually depends on the initial conditions. So if you start at 50-50, maybe the orange beats the blue, but then if you start at some other initial state, the orange ends up going extinct. And so again, the point here is that all of these things can arise just from these very simple features of microbial growth that I showed in the model a couple of slides ago. And they all really are possible because it's this growth that's happening out of steady state. And really the underlying feature that causes sort of this non-steady state behavior to work this way is because there are trade-offs in these growth traits. And just to give you an example of how this works, if you look at the growth rate and the lag time of two different strains, a trade-off would mean if you have two strains that are basically along like a diagonal line here. So say this blue point here has slower growth rate than the other strain, but a shorter lag time. So it's better in one thing and worse in the other. And it turns out that that particular combination is what's necessary to get stable coexistence of these strains. So the blue lines here represent just two trajectories from initial conditions of how this particular strain would behave relative to its partner. And you can see that in this case, it stably coexists at 50-50. And the reason why this happens in this particular case is that, so I told you this blue one has a slower growth rate, but a shorter lag time. The other key ingredient is it also has to have a trade-off in its yield, its resource efficiency. And in this case, it has to have a higher yield than the other strain. And so what happens is then when this blue strain is rare, the overall yield is low because it's sort of dictated by the other strain that has a lower yield. And that favors this blue strain because it has a shorter lag time, because lag time is more important if you have a shorter competition with a smaller fold change. And that's what allows it to invade. But then when it's abundant, as opposed to being rare, it's a disadvantage because now the yield is very high and it has a slower growth rate. So that's bad for it. And so that's what causes it to have this stable coexistence. And there's sort of an opposite argument that it would allow you to have this bistability regime that I showed on the previous example. So, but here the point is that all of these things are underlined by some kind of trade-offs in these growth traits. So then the empirical question for the biology is, of course, are these growth traits actually realized? So I told you about what's possible in a model, but do we actually see evidence of this? And as is often the case when you ask these sort of empirical questions, the answer is sometimes. And so we were originally thinking about this motivated by a collection of E. coli strains in my postdoc lab that had mutations in this particular protein. That's not really important for now. But if we looked at the growth rates and lag times of all of these different mutant strains, we saw that while there wasn't a trade-off, overall the strains, we definitely found certainly some pairs of strains, like say one over here and one over here, that do have a trade-off between their lag times and growth rates. And we were able to actually verify that was realized in their dynamics when we competed them together. So there are certainly examples of pairs of these, if not statistical trends. But people have seen interesting examples of this in many other systems now too, which I think is very exciting. So there's a paper that many of you may be familiar with that Marcus Bassan did during his postdoc with Uwe Sauer and Terry Hua. And what they found was actually a trade-off between growth rates and lag times, not between strains, but across environments involved in shifting between different carbon sources. So I want to emphasize this is actually a quite different kind of trade-off than what I'm talking about here, but there's potentially relationship to it, which has been more recently published, but I won't talk about for the sake of time. And then I think one of the most compelling examples is a couple recent papers from Bloxham and Jeff Gore's lab, where they found pairs of these soil bacteria that had trade-offs between how fast they grew sort of on an initial carbon source and how quickly they were able to switch to a second carbon source afterward. And they found that this was certainly consistent with the coexistence behavior they saw between these species. And if anyone's interested more in kind of this sort of literature on trade-offs, Eustace and I just wrote this opinion article where we've tried to really categorize these things and document them more carefully, because of course there's a lot of different things to talk about here. But I would say the current state of this evidence is that there are certainly some evidence, some examples of these trade-offs, but it's not clear whether these are common or really a dominant mechanism for generating these kind of dynamics in nature. So that's still, I think, very much an open question. Okay, So now I want to move to the second question about how microbial growth response evolves according to the environment. And the motivation here is that so far what I told you is assuming that basically growth happens at a constant rate as long as this resource is present, but regardless of its concentration. Of course, in microbiology, we've known that that's not true. And this is some of the most classic work in microbiology dating back to Jacques Monod, where if you look at the growth rate of some population as a function of the concentration of a resource, like say glucose, you see that that growth rate varies quantitatively over that concentration range. And at very low concentrations, you tend to have sort of an approximately linear dependence. But then once that concentration gets higher, this starts to level off and saturate at some maximum growth rate. And mathematically, this has been most famously parameterized by this Monod model of growth rate. And here's the equation for it that I'm going to be referring to. And so this is how growth rate is a function of a resource concentration R. And you can model it as this hyperbolic function where you have some maximum growth rate, G max. And so that's the sort of saturation growth rate you would have up here when you have unlimited amounts of this resource. And then there's this other key parameter that's usually called K. And sometimes this is called affinity, but actually I think affinity is a misleading word for this because it's kind of reciprocal to infinity. So we call it the threshold resource concentration. And it's basically a concentration of resource down here where basically the growth rate starts to really be sensitive to the resource concentration. And that's why I think, threshold is kind of a good term for it. But you should think of it as it's a concentration of the resource. And so it tells something about where the growth rate is really going to be sensitive to that particular resource's concentration. So these are these two key parameters of this dependence. And one of the things we wanted to do is first find out what is just the biodiversity of these parameters, because, often, let me just go back for a second. We write down models of these sorts of things, but we often don't really know what are reasonable values of these sorts of traits that we should be thinking about in these models. And so Eustace and Noel did this really heroic effort of going through literally decades of papers and trying to find good measurements of these parameters across many different organisms and many different nutrients. And it has to be decades of papers because for various reasons, people don't measure these things very much anymore, basically because I think there's no omics measurements for it. And so this is a very kind of classic microbiology thing that, you really have to look back at more historical work for. So that's very difficult, but we were able to get somewhat over 200 different data points. And this shows a summary of the data that we found for this resource threshold K. This is in micromolar here. And here it's organized by all the different nutrients we found. And then the color coding tells you different organisms. So some were prokaryotes, some were eukaryotes, some were autotrophs, some were heterotrophs. And I think the salient feature here that I want to emphasize is that you can see there's a really wide range of these thresholds K, even for the same nutrient. So for instance, if you look at glucose here, there's orders of magnitude differences between some of these thresholds, that are less than a micromolar, and then other ones that are, over 100 micromolar. And that's true for pretty much all the nutrients that we have significant data for. We also did this for these maximum growth rates, the Gmax, and these certainly also have quite a bit of a range, but it's a lot smaller. This is also a log scale, but it's not nearly as big. And generally, the structure here is that we see sort of these growth rates kind of cluster according to whether you're a heterotroph, where these maximum doubling times tend to be around an hour, versus an autotroph where these maximum doubling times tend to be around 24 hours, which I think that part's certainly not surprising. I think the hour part is kind of a little interesting, but here there's kind of more of this bimodal shape between these parameters, just depending on kind of the energy metabolism of the organism. Okay, so the question though is, how would you possibly describe or what sort of explains the overall diversity of all of these traits? And going back to just what I talked about in the first part, One of the main hypotheses here that's been around for a long time is whether there are trade-offs between these traits, between the maximum growth rate and the resource threshold, and whether that's, kind of the important driving force that explains this diversity. And what a trade-off looks like here is that you would have one strain, say the orange one, that has a very low value of the threshold K, meaning that it can grow very quickly at low amounts of resource, but then it has a lower maximum growth rate, meaning that it sort of levels off at a kind of a slow amount of growth, compared to another strain, green, that has a higher K, so it's not as good at growing in low resources, but once the resources, get very abundant, it can grow a lot faster. So people assign these archetypes of an opportunist and gleaner to these two strains to sort of characterize these sort of two different strategies of growing fast when resources are scarce versus growing fast when resources are abundant. So this is a hypothesis, and the question is this really true in the data? And we find just no evidence that this is true. And I think the best way to look at this is if you look at a single organism across and a single resource at a time. So for example, the green points here, these are all E. coli on glucose. The horizontal axis you see is the maximum growth rate. The vertical axis is this threshold K. And you can see that there's really just no clear trend here. If you look at the statistics, there's sort of a positive correlation. So here, this trade-off, again, is a positive correlation. So higher maximum growth rate would mean a higher K. But, this is nowhere close to being statistically significant. It's very much driven by a couple outlier points. And then actually, if you look at yeast on glucose, you see the opposite trend. You see something that's negative, which instead of being a trade-off really means there's like a synergy. The strains that are faster at growing at low amounts of glucose or even faster at high amounts of glucose. And we looked at this over other nutrients that we have in the data, so phosphate, nitrate, silicate, and you just don't see this in any of these cases. Silicate also actually has this like, synergistic rather than trade-off trend. So in this data, which as far as we know is really the most systematic collection of these measurements that's been put together, there's just no evidence in any of cases for systematic trade-offs in these traits. So some of you who may be familiar with this literature may be wondering, well, wait a minute, people have talked about this a lot. People have claimed to find other evidence of this. So what's the source of this inconsistency? And basically, we argue that the reason this has been very confusing historically is that people are conflating population growth with resource uptake. And I think there's a very good reason for why this is confusing, because the mathematical models of these two things are essentially equivalent. So when we model resource uptake, we usually model that as a Michaelis-Menten process with some maximum rate Vmax and then some half saturation constant little k. And as you can see, this is mathematically equivalent to this Monod model of growth that I've been talking about for the last few slides. But the thing is that these are mathematically equivalent, but they're not biologically equivalent. So uptake is certainly related to growth, but it's not all of growth. Uptake is the first step in some process that ultimately leads to the growth of new biomass, but it's not the whole thing. There's many other steps that come afterward. And so I think it's a pretty big stretch to imagine that what you would see in uptake completely determines what you would see in growth. And just to make this more explicit, some of the data we have also has sort of corresponding measurements of uptake rates for the same nutrients and same organisms. In this case, these were all measurements for phosphate, and I think these are all phytoplankton. And you can see here on the left, this shows the threshold K for growth rate for phosphate. The vertical axis is the constant little k for uptake. And you can see that there's no correlation here between uptake and growth rate. And if you look at the sort of maximum rates, the maximum growth rate versus the maximum uptake rate, there's sort of a correlation, but again, it's very noisy around it. And so I think, it's pretty clear from looking at these plots that even if there is a trade-off among these uptake traits, that's not going to be translated into a trade-off among the growth traits. And so this is why there has been a lot of kind of confusion about this historically, and why there could be trade-offs in the uptake, but not in the growth. So if there aren't any trade-offs, what does determine the diversity of these Monod growth traits? Obviously we see a lot of diversity. And in particular, another hypothesis that has been discussed a lot in the literature is the relationship of these thresholds K to the environmental concentration R of that resource. And in particular, a lot of people either explicitly or implicitly assume that these thresholds are proportional to the resource concentration. So if you have a more amount of the resource in the environment, then this threshold K is going to evolve to be higher proportionally. And one of the reasons this would be a very useful thing, if it were actually true, is that it would mean you could infer environmental niches from these traits. So as I mentioned a little bit at the beginning, measuring growth and things about nutrients in these natural environments is very difficult. So it's a lot easier if you can just measure traits of growth for these organisms and then use that data to infer something about the natural environment from it. And so if you could then just measure, say, these K thresholds across a bunch of different organisms, so like this was an example across a bunch of marine species a while ago, you could then say, some of these have very low Ks, some of them have high Ks. So that must mean the concentration of the resource, which was ammonium in this case, is proportionately higher for these guys in their environment than for these guys. But this is an evolutionary hypothesis. And this is something, therefore, that needs to be tested, using sort of an evolutionary process. Doing evolution experiments would be great here. These are potentially not practical given the time, range that you would need to actually see this. But that makes it perfect for testing, at least through modeling, to really see if the fundamental principle actually works. So that's what we did. And we constructed this model that's kind of similar to the dynamics that I showed you in the first section about the model. So we have some population that grows according to these Monod dynamics, where it's given a supply of the resource, it grows, and then according to this Monod model, the growth rate sort of is fast at the beginning when the resource is abundant, but then slows down until the resource runs out and it reaches this saturation. And we can imagine that there are now these mutations that arise that produce small perturbations in these traits, the maximum growth rate Gmax and the threshold K. And now these mutations then either fix in the population according to some model of population genetics, or they go extinct in the population. And we can imagine then a serial process of these mutations accumulating that ultimately determines an evolutionary trajectory of these traits. And one thing I want to emphasize here is that it turns out that a small detail in how we model the population dynamics over these cycles of growth turns out to be really important. And so that's why I'm showing it here. So as I said before, we model sort of the long-term dynamics through these batch-like growth cycles where there's sort of resources, growth, and then dilution, and then the cycle repeats. But how exactly this bottleneck happens turns out to be important. And so one way you can do this is that after growth has stopped, when the dilution happens such that the population always goes down to a fixed biomass at the bottleneck. So maybe it goes up and then it's diluted down to 1 million cells. And it goes up and again diluted down to 1 million cells. But another way you could do this is instead of diluting down to a fixed bottleneck, you could dilute by a fixed factor. And you just say, okay, after growth happens, you just dilute 100-fold every single time. And this is actually how most people would do it in the laboratory because it's easier. But as you can see there, there are slight differences between these two things, because if you have a fixed bottleneck, you're always going down to the same size. But if you have a fixed dilution factor, you're not necessarily going down to the same size because you're just diluting by 100-fold. And so that depends on how much growth actually happens during the growth cycle. But as I'll show you, this turns out to be an important difference. But before I get to that, what just happens overall with the adaptation of these resource thresholds? So like I said, we can simulate this evolutionary process where the population, say, starts at some point here with some relatively high value of this threshold K, which means it grows, it slows down a lot when it runs out of the resources. And not surprisingly, over evolutionary time, which we measure as the number of sampled mutations that accumulate, this threshold K goes down because lower K means faster growth. And so there's positive selection for that to happen. So it goes down a lot relatively quickly. But what I think is especially interesting here is that you can see that it doesn't just keep going down forever. It goes down, but then really dramatically slows down its decrease over evolutionary time. And just to give you sort of a more biological sense of what this amount of change in this threshold K represents, If you look at the beginning here, the threshold is about equal to the concentration of the resource, and you would get a growth curve that looks something like this, with a very smooth transition between exponential growth and saturation. By the end, it's 1,000 times lower. And in that case, you end up having this really abrupt transition from exponential growth to saturation. So this is a really big change in dynamics that occurs. So why does it slow down, though, and sort of reach approximately this kind of limiting value? And the reason is that, well, as I just mentioned, obviously there's selection here that's driving K downward, because lower K is always better. But the thing is that selection has some diminishing returns. So selection is very strong when you have a growth curve like this, because reducing K a little bit is going to have a big impact on what these growth dynamics look like. But once K gets really low like this, there's not a lot more room to go here. This is already such an abrupt transition, and reducing K by another 10% or something isn't going to really lead to much difference in these growth dynamics. So selection starts to get very, very weak as K decreases. But of course, selection is not the only force driving evolution of this threshold. There's also genetic drift, among other things. And genetic drift here represents the demographic fluctuations that occur in this population owing to a finite population size. And these fluctuations are constant over this range. So, even up here, they're sort of fluctuating around, but then down here, it's the same. And so what happens is that once K gets low enough, the strength of selection gets so weak compared to the strength of genetic drift that basically selection is no longer able to efficiently reduce K anymore. And that's why you start to get sort of this leveling off in the threshold. And so you can quantify this as the point where the strength of selection is approximately equal to the strength of genetic drift, which is parameterized as the reciprocal of this effective population size. And this condition is called selection drift balance. And so what we can do then is we can basically, because this is a relatively simple model, calculate this selection drift balance condition and determine where this evolved threshold, this evolved value of the threshold occurs as a function of, say, the resource concentration and the other underlying parameters in the environment. And it turns out there's a relatively simple answer here. And if you look at the evolved threshold K, it's proportional to the resource concentration divided by the effective population size. And I think superficially, this looks, incredibly intuitive. So on one hand, you see that, yes, it is proportional to the resource concentration. So if you double the resource, you will double K. And then it's inversely proportional to this effective population size, which also makes sense because if you have a bigger population size, genetic drift is going to be weaker, and therefore selection optimizes more efficiently, and you're going to be able to evolve to a lower value of this threshold K. So it turns out if you're in this fixed bottleneck regime of the population dynamics, that everything I just said is exactly the whole story. So you can look at, say, the evolved threshold as a function of this initial resource concentration. And the blue line here is this fixed bottleneck regime. And you can see, yes, it's pretty much just proportional. So if you have more resource, you're going to have higher K. So then this whole sort of inference idea of measuring traits and inferring the natural environment, that works. However, if you have the fixed dilution factor regime, then this breaks down. And the reason is that this is still mathematically true, but the problem is that there's a hidden dependence of the resource concentration in the effective population size, because the effective population size under these dynamics is basically the size at the bottleneck. And if you have a fixed dilution factor, that bottleneck size is not independent of the resource concentration, but rather it's proportional to it. So if you double the amount of resource, you end up doubling the bottleneck size because you get twice as much growth, but then you still only dilute by a factor of 100. And then you just go down to a higher bottleneck after this cycle occurs. And so what happens is that you have proportionality to the resource here, but you also have proportionality to the resource down here. And then that cancels out. And then you have an evolved threshold that's approximately independent of the resource concentration. And so what that means in that case, under these dynamics, is you really can't learn anything about the resource concentration in the environment from looking at these thresholds. You could basically have any concentration of the resource you want, and you could still get the same or different values of the threshold. And just to kind of give you an example of this, this plot here is calibrated to E. coli data for glucose. And you can see on the sort of right, this distribution of these measured thresholds for glucose. And you can see that basically if you had these fixed dilution factor regimes, you could have like any concentration of glucose in the environment that you'd want, and that would be consistent with this whole range, which is orders of magnitude of these thresholds K. So one of the more recent things we did was try to actually collect some environmental data on these natural concentrations for the strains that we had, good quality measurements of the thresholds for. And this plot is, it's pretty limited data, but this is what we were able to collect so far. This shows this resource threshold K on the horizontal axis compared to some very approximate environmental resource concentrations R on the vertical axis. The colors represent different nutrients, but that doesn't really matter for right now. But what you can see is that there is some correlation across some orders of magnitude. It's a lot of variation around it, but there is some correlation. And so this does suggest that maybe it is more like the fixed bottleneck regime rather than the fixed dilution regime. But there are certainly other mechanisms that could be important here that I won't talk about now, but I'm happy to discuss in questions later if anyone's interested. Okay, so in the last few minutes, I just want to very briefly talk about this last part of the talk, which is about the possibility of co-limitation by multiple resources. And I mostly just kind of want to convince you, I think this is a very important aspect and underlooked aspect of microbial growth. So far I've talked completely about models and data, assuming there's a single limiting resource. But we know that microbes depend not on one, but on many different resources. And I think it's really helpful just to look at the periodic table to see all the different elements that microbes need to grow. And of course, these all don't come in one super molecule. You have to get a lot of different nutrients to get all of these different elements. And this is, I think, particularly acute in ocean environments where people know that many of these different elemental resources are rare. And in particular, some of them are simultaneously rare. So like especially some of these metals like cobalt and manganese and iron. And so why would you care about this resource limitation? And I think the most practical setting in which you'd care is for controlling microbial growth. So you could imagine if you had a population where you knew there was just single limitation for one resource, like say carbon. Now, if you take this population and you add carbon to it, you're going to see more growth. And if you add something else, like say nitrogen, you're not going to see growth. So now if I want to make growth happen, I just add carbon and not anything else. But if you have co-limitation, this is also really important to know, because if you just added carbon, but you're co-limited for carbon and nitrogen, you're not going to see any growth. You need to add carbon and nitrogen simultaneously in order to see growth happen. And there's an analogous version of this for trying to stop growth. Like if you have, instead of a commensal or a probiotic, like here, you would have like a pathogen that you want to stop growth. You also need to know what it's co-limited for if you'd want to be able to inhibit that growth, because taking away one thing, may not have the effect that you want. So one of the main things that we've thought about here is really how to quantitatively describe limitation rather than thinking about it in sort of these very qualitative terms like, adding something and seeing growth or not. And we focus on a particular concept that we call the limitation coefficient. And mathematically, this is defined as the relative change in growth rate due to a relative change in the resource. And you can define this through this derivative of the growth rate. And what I think is important here is that now if you look at, say, again, like a Monod-like dependence of growth rate on resource concentration, and you can then calculate, at least either in a model or using real data, what this limitation coefficient would be. And as you can see that it's not just 0 or 1, it's actually this whole continuum of states. So instead of having just, resources limiting when it's rare, resources not limiting when it's abundant, you have this continuum of limitation that can range all the way up to some maximum value when the resource is very rare, and then smoothly varies down to a minimum value when the resource is abundant. So it's not just on or off, but it's really a quantitative number that characterizes the state of a population. And in particular, it's a number that therefore can change. because it depends on the resource concentration, and if the resource concentration changes over time, as it always will, this limitation state also changes. So this is really an instantaneous property of a population as well, not something that's sort of just true over, an arbitrary window of time. Finally, how can we talk about co-limitation? Well, we can take limitation coefficients for all of the different resources we have in the environment, and we can take this kind of average of them, and this defines a quantity that we call the effective number of limiting resources. And we call it this because it ranges between a minimum value of 1 if all of these limitation coefficients but 1 are 0, and then you basically you have one non-zero number here plus a bunch of zeros, and then you divide it by the same number, and so you get 1. And therefore, that's what you expect, right? There's only one thing that has any limitation, and so the number of limiting resources is therefore 1. Whereas if you had a bunch of resources and their limitation coefficients were all equal, then this effective number turns out to be equal to the actual number of resources you have in the environment, because you're adding up all the same limitation coefficients here. And so this tells you, therefore, some, again, quantitative measurement of the number of limiting resources that doesn't just have to be some integer value of 1, 2, 3, 4, but you could have 1.8 limiting resources or 2.7 limiting resources, which captures the fact that you have some spectrum of limitation coefficients across all of your different resources. And just to give you an example of this, if you look at, say, some two-dimensional space of resource concentration, so you have resource 1 here and resource 2 here, you can look at the number of limiting resources as a function of these concentrations. And you can see that, if you're up here where you have a lot of resource 2 and a little bit of resource 1, you have approximate single limitation for resource 1, and then vice versa in this corner. And then in the middle, when you have sort of approximately equal amounts of the two resource, that's where you would achieve co-limitation, and you have approximately 2 limiting resources. And so altogether, this is, we think, a really convenient way to quantify these questions about limitation that you can therefore apply to experimental data as well as thinking about in the context of models. So for the sake of time, I just want to emphasize kind of one aspect of this, which is, can we actually measure co-limitation in the way I've talked about under laboratory conditions? And the answer is yes. And the sort of way we set up all of these experiments is we take 2 different resources and we vary them over some 2-dimensional gradient, and we measure growth at all combinations of these resource concentrations. And altogether, we get a landscape that looks like this, where we have some output of growth, like growth rate, as a function of 2 different resources. This example is from E. coli on glucose and ammonium. And you can see that you can measure this data and then fit some model to this. And then from this model, then you can then precisely calculate these limitation and co-limitation conditions. And so, for example, here on the left, this shows the 2-dimensional space of glucose and ammonium concentrations. And here we have just labeled where these different limitation regimes occur. And so the blue contours indicate different amounts of limitation for glucose. The orange indicates different limitation coefficients for ammonium. And then we can use that to then calculate the co-limitation between glucose and ammonium. And you can see that there's this quite wide range, actually, of conditions where you would have significant co-limitation for glucose and ammonium. And what I think is especially interesting here is you see how much this overlaps with what's estimated to be a natural range of concentrations for glucose and ammonium for E. coli. And that suggests that they may often experience, in fact, these co-limitation conditions in nature, not just the ones, we have sort of engineered in the laboratory. And one of the things we're doing with this that I think is going to be really interesting is now this allows us to, precisely map out the space of limitation conditions quantitatively, and we can then construct very specific states of limitation and then go test other aspects of physiology in those conditions. And so, for example, we can construct states of single limitation for glucose and ammonium, and then also co-limitation, and then start to look at the physiology of cells under those conditions and see to what extent they're different, and what are the actual mechanisms that cause them to be limited or co-limited in these different ways. So I think I want to stop there, and I will skip these last few things. but of course, I'm happy to answer questions more about the co-limitation work that we've done here if there's any interest. But I want to just make sure I give you a summary of all the things I've talked about so far today. So in the first part, we talked about how does non-steady state growth of microbial populations affect evolution? And here I think the point is that when you have growth trade-offs, this readily produces, under these non-steady state dynamics, complex selection forces like non-transitive selection and higher order ecological effects. But the evidence for this empirically is still, I think, very much an open question. And the second part, we talked about how the microbial growth response to a resource evolves to be commensurate with that resource's environmental availability. And as I showed, there's huge amounts of variation in these growth response traits across resources and across organisms. But we find that this relationship between those responses and the environment is complicated, and it depends crucially on the mode of population dynamics. For example, whether there's a fixed bottleneck or a fixed dilution. And we would really therefore need to know more information about those population dynamics in these natural environments in order to make this kind of inference. And so you can't just, naively apply this to the data you would observe. And then finally, is microbial growth co-limited by multiple resources? We think the answer is yes. We've certainly been able to engineer this in the lab. I didn't talk about evolution, but we think also evolution will often make this so. And this also has interesting impacts for ecological interactions and cross-feeding, which I haven't talked about, but I'm happy to discuss with anyone who's interested. And finally, as Marco mentioned, I recently moved back to Rutgers here earlier this year, and we're recruiting for different positions, postdocs and PhD students. And so if you're interested in any of the kind of work I've talked about today, or some of the other things we do, like the effect of cross-feeding on mutations and adaptation, please don't hesitate to get in touch with me by email or check out our website. And I'm happy to answer any questions or discuss any comments you guys have. Thank you so much.
Thank you, Michael, for the talk. So now we have a few minutes for the question. I'll stop recording also.